Good morning, Dr. Soshnev. Can you tell us how you first got an interest in neuroscience and this field in general?
Ooh. Science in general, I guess, all kids like bugs and dinosaurs. And
Hmm.
when I was 10 years old, I collected bugs, and then one thing led to another.
Hmm.
I don't think there was a particular moment.
Hmm.
Just kind of like how things work is always
Hmm.
fun. I'm not smart enough to study math. I am not like technically gifted enough to study physics, and so
Hmm.
it ended up being life sciences.
Hmm.
Um, Neuroscience, that's a little complicated. I guess there were a couple moments in my career. Okay, way back when, right, when I was a grad student and I didn't know what I wanted to study. This
Hmm.
was early, late 2000s.
Hmm.
Fire and Mello just won the Nobel Prize for small RNAs. I wanted to study
Hmm.
small RNAs. That sort of pushed me a little bit into neuroscience because microRNAs became kind of hot in gene regulation in CNS.
Hmm.
Um, I ended up working on something else during my grad school, but we had a curious phenotype about how germline, which like I don't know if you guys know what germline is, but it's the cells that contribute to sperm and egg development. It's basically like probably the most important lineage in
Hmm.
biology because that's what
Hmm.
gets propagated through generations. So germline um, is actually surprisingly um, connected to neural fate. During very early egg and sperm development, neuro gene expression has to be tightly controlled and suppressed because if it isn't, then essentially you have ectopic expression of neuron-specific programs in the germline, which is kind of cool.
Hmm.
So that was basically the take home of my 6 years of PhD thesis. That was fun.
Hmm.
And then I went on as a postdoc to work on just more complicated gene regulation stuff,
Hmm.
more mechanistic gene regulation stuff.
Hmm.
And that's when I learned a really interesting fact about neurons. Well, I kind of knew that, but I mean, you probably know
Hmm.
that from like reading regular textbooks
Hmm.
that neurons don't divide,
Hmm.
right?
Right.
But when you kind of think about how most cells divide, but neurons
Hmm.
don't, you realize that the proteins that organize the DNA in the neurons have to be very, very special because these proteins essentially have to live for a very long time
Hmm.
and they have to be very stable and they have to be very stably incorporated and maintained. So where regular cell, I don't know, in tissue culture, regular cell divides every 24 hours. In human body, there are some cells that divide like every 8 hours, very,
Hmm.
very fast.
Right.
They have a chance to renew their
Hmm.
nuclear content because every time you divide, you have to kind of like make new stuff.
Hmm. Right.
Neurons don't make new stuff.
Hmm.
And so they accumulate errors, they accumulate problems over
Hmm.
time.
Hmm.
We're now interested in that, how that connects to neurodegenerative disorders. We have been interested in just basic mechanisms of that for a long time now. So
Right.
yeah,
Right.
that's the long story.
Hmm.
Mm-hmm.
And of course, I know when getting to your position like this, you've obviously come against like some troubles or problems. So, do you have like a moment in time where you were stuck at like a very like hard point in your research or whatever?
Scientifically,
Yes.
I don't know about stuck. I can tell you that it's very rare for people to work on one thing.
Hmm.
And so, yes, things do get stuck in a sense,
Hmm.
but also because you work on more than one thing at a time, when one thing slows down, another thing picks up, you
Oh,
can
I
kind of
see.
juggle your attention a little bit.
Hmm.
It, in general, science doesn't work most of the time, right?
Hmm.
There is this statement that people say very often, and I kind of get irked by that when they write a grant and they say, we will confirm our hypothesis. And that
Hmm.
always rubs me the wrong way because we don't confirm anything,
Hmm.
we test
Hmm.
things.
Hmm.
We come up with ideas, we test these ideas. These ideas don't work most of the time,
Hmm.
and there's
Right.
nothing wrong with that.
Right.
That's why, by the way, science is a non-profit by definition, because if you try
Hmm.
to make profit off of science, like you're just gonna go bankrupt in about 6 months.
Hmm. Yeah, right.
Um, okay, so of the experiments that kind of, make me sad a little bit, well, not sad, but like definitely cost a lot of money and didn't
Hmm.
lead anywhere.
Hmm.
In my grad school, there was a choice. We were studying gene expression in like a specific tissue. We were specifically interested in, the whole transcriptome. So I don't
Hmm.
know again how much you know about what the genome looks like. But imagine, you pull out a cell and you have this collection of RNAs,
Hmm.
which corresponds to specific genes. And you want
Hmm.
to quantify these RNAs all at the
Hmm.
same time.
Hmm.
And nowadays, this is a bit of a piece of cake because technology has evolved. You can do something called RNA-seq, and
Hmm.
we do that all the time in the
Hmm.
lab now. But when I was in grad school, that was like year 2008,
Yeah.
2009, we turned to this platform, developed a chip with a company called Agilent, um, and collaborators. We weren't the ones developing the actual chip. And after spending a year and lots of effort, it
Hmm.
basically didn't work because we
Oh,
just didn't have the analytical pipelines. We saw the data, but we couldn't put numbers on the data,
I
which was
see.
really kind of annoying.
Hmm.
We still got some info from it, was still interesting, but we had to switch the technology.
I see.
And that probably set me back by a year in grad school.
Hmm.
In postdoc, I made a mouse from scratch, doing gene editing in cell
Oh,
cultures and making a mouse out of that,
oh
with the
wow.
idea to, I mean, that's pretty standard tech.
Hmm.
I was thinking that we could do some behavioral assays because we introduced a mutation that is known to cause a neurodevelopmental disorder in humans into a mouse embryonic stem cell culture, then grew mice out of those cells, which is kind of magic, to be honest. It's a lot of fun. But then these
Hmm.
mice turned out to be sterile.
Oh.
And that kind of killed the pipeline because you can't breed them and you can't have controls. Like, again, all
Hmm.
experiments need controls,
Hmm.
and
Right.
controls are frequently more important than the experiment itself because
Hmm.
without the control, you can't interpret the results.
Hmm. Right.
And so we couldn't breed these mice. And that was, again, many tens of thousands of dollars and many, many months later.
Hmm.
But it's one of those things where, like, we didn't know.
Right. Right.
Again, at the same time, I was doing other things. So it
Hmm.
was never like, oh, the only thing I'm doing is sitting in my butt for 2 years and waiting for the mice to come of
Right.
age.
Right.
Like, right.
Um, do you teach more often or do you do research more often these
I do
days?
research. I do
Hmm.
research.
Okay.
I teach a little bit,
Hmm.
and I enjoy teaching very much. It's a very different type of activity.
Hmm.
I am what is called research active faculty,
Hmm.
where basically my job description stipulates that my research effort is significantly higher than my teaching effort.
Oh, I see. I see. I see you have a publication called The Conserved N-Terminals SANT1 Binding Domain of
Oh yeah.
EZH2 regulates PRC2 activity. How did you come up with this research, or if it was collaborated, and how, like, did you
Wow.
perform the research?
It's funny that you found that one. That's brand
Mhm.
new.
Yeah.
Okay, so that was actually, that's a really interesting story. I was making a, again, I don't know how much you know about Polycom. Do people, so
Not
let me take a step back. People watching this, do they know anything about Polycom? Who is this for?
just like a broad audience of like,
Okay.
yeah.
All right, so I have to take a huge step back because we're not going to talk about sand domain before, or sand binding domain, I should say, before we talk about polycomb.
Hmm.
So, it's have you ever thought about how, we have two arms and two legs and,
Hmm.
a certain number of ribs,
Hmm.
and the snake has this like 1,000 ribs and
Hmm.
like 1 million vertebrae, but no
Hmm.
arms,
Right.
and a fly has 6 arms, like 3 pairs of arms,
Hmm.
basically. But
Hmm.
each arm is doing its own thing, right? Like
Hmm.
the hind legs are not like the front legs.
Hmm.
So very long time ago, there was this guy named Ed Lewis, and he
Hmm.
had a wife who was also a brilliant scientist who doesn't get enough credit for her contributions, really. But they investigated the regulation of a gene cluster called HOX,
Hmm.
mutations in HOX genes called homeotic transformation. Like just Wikipedia this. I'm trying to cram the whole development of biology into 1 minute
Yeah.
here.
Hmm.
So he discovered that Hox locus, which is in fruit flies on a certain chromosome, I don't remember off the top of my head where, it's about 100 kilobase locus, contains many, many genes
Hmm.
arranged linearly,
Hmm.
and certain genes turn on in certain body segments.
Oh. Hmm.
So that like abdominal segments turn on the very end of the Hox locus, and then as you get closer to the head, you progressively turn on other Hox genes.
Hmm.
Eventually, Ed Lewis figured out the logic for how Hox is regulated
Hmm.
and what specifies these master genes that tell the body segments to make either the abdomen or the thorax or the legs or the wings or the head.
Hmm.
And what's absolutely fascinating about this is turned out that Hox genes are extremely conserved, and humans have Hox genes too.
Hmm.
And the arrangement of Hox genes in humans is the same as in fruit flies and the same as in worms.
Oh.
And the mechanism that regulates Hox is also incredibly conserved.
Hmm.
It is established by an enzyme complex called polycomb, which
Hmm.
takes its name from fruit flies, because if you mutate a specific subunit of ERC1, you have what's called sexcom transformation on the legs. Like basically your hind legs become like your front legs.
Hmm.
The only person who could see that was Ed Lewis's wife, because
Oh.
it's very, very, very hard to see.
Hmm.
Eventually, they came up with much cooler homeotic transformations, like trithorax, for example, where a fly grows extra pair of wings.
Hmm.
Um, but essentially, what I'm trying to say here is that body segmentation and the body plan,
Hmm.
like your front to back body plan,
Hmm.
is established through an extremely conserved mechanism that is the same in every multicellular organism.
Hmm.
The enzyme that regulates Hox genes, it's called polycomb, it's a large complex.
Hmm.
And the key subunit in it, in flies, it's called EZ, enhancer of zest.
Hmm.
And in humans, it is called EZH2, enhancer of zest homolog 2.
Hmm.
How EZH2 is regulated has been subject of study for like many, many years, many, many people, because it's central to development. It's like one of these critical, critical components of development. By the way, Ed
Hmm.
Lewis won Nobel Prize for this. So
Oh.
Google him. It's important. Okay,
Hmm.
this is not like some random science. This is really,
Yeah.
really important stuff.
Hmm.
If you take college level developmental biology, you will spend weeks talking about this. This is
Oh.
fundamental.
Hmm. Hmm.
Um, anyway, oh yeah, and the fun fact is he figured all of that out before the whole genome sequences were available. So
Wow.
all of that was pure genetics and pure logic. Like his Nature
Wow.
paper from 1970s was it's a masterpiece. Like look it up.
Hmm.
It's fascinating.
Hmm.
Anyway, so a while ago when I was in my postdoc, I was making a knockout of an enzyme called BZH2 in
Hmm.
mouse CS cells. We often use mouse embryonic stem cells for studies of this complex because polycomb is very important in ES cells. It has observable phenotypes.
Hmm.
And uh, we use CRISPR for this. CRISPR is kind of like a
Hmm.
everybody likes CRISPR, but it's
Right.
a tool that is very, very easy to work with these days.
Hmm.
We used CRISPR for this, and we essentially accidentally made a small deletion in the first 20 amino acids of EZH2,
Oh.
which killed the first 20 amino acids. But just serendipitously, there is a methionine in position 21 or 23. I don't remember what number, but like right after, there
Hmm.
is a new methionine. And if you remember your molecular biology, methionine is an ATG. ATG can be a start codon. It is
Hmm.
a start codon. So
Hmm.
even though we removed the original start codon, we now are translating from the second methionine downstream.
Hmm.
So we created an EZH2 that is essentially lacking first 20 amino acids.
Hmm.
And it turns out that it had really interesting phenotypes.
Hmm.
And the interesting phenotypes were that despite the fact that EZH2 is a large protein, the enzymatic component is here, like very,
Hmm.
very far away.
Hmm.
We lost very, very small chunk far down here.
Hmm.
But this mutant EZH2, even though it is targeted to the genome accurately, it is normally processed, it's accumulated at normal levels, it is almost completely dead enzymatically.
Oh.
So somehow it's like you get a scratch in your windshield and like your car doesn't run anymore.
Hmm.
Like that type of analogy, right?
Hmm.
So that was it. And then
Hmm.
we wanted to figure out what it does, and we spent a lot of time trying to figure out what it does. That story was really done by Agata Patriotis. She is now in Tyler Jack's lab at MIT. She's a
Hmm.
postdoc there.
Hmm.
But I generated the initial mutation, and then I worked with Agata to
Hmm.
kind of put the story together. She did most of the work on that.
Hmm.
Like I just helped here and there a little bit.
Hmm. And
Mhm.
of course, you are, and were you like doing other research as well
Of course.
simultaneously? Yes.
Yeah, yeah, yeah.
Hmm.
That was never my main project. It's
Hmm.
just it took a long time to shepherd that story to publication.
Hmm.
My main project from that period is a collaborative paper with Ari Melnick and Ethel Caesarman about the role of linker histones in hematologic malignancies. That
Hmm.
was published in Nature in 2021, I think.
Hmm.
Yeah,
Also, there's another publication I want to like just like ask about, which is RNA seq dataset of wild type and
yeah,
PRC2 mutant
that's
mouse.
just
Yeah. Is
that's
that
just
like
data
another
set,
side project of yours?
pretty much. Yeah.
Hmm.
So again, I think publication like there are different types of publications, right?
Hmm.
There are publications where you're presenting new ideas, new research, like
Hmm.
really wrap it up as a model, as a story, a research
Hmm.
paper. So the genes and development paper we were talked about previously, that's one of
Hmm.
those papers.
Hmm.
There are some publications where you just present the methods, or you
Hmm.
present, you just want to throw something out there for the community to look at, and maybe somebody
Hmm.
will find something interesting for them.
Hmm.
That is, that's that one.
Hmm.
To be completely frank, and again, I don't know who's going to be watching this. There is this mantra about publish or perish,
Hmm.
where we're basically expected to publish stuff.
Hmm.
It has been both good and bad for science because, good thing that, there are some deadlines and you got to publish stuff. The bad thing is that sometimes it forces you to publish things that are maybe not ready for prime time,
Hmm.
and you just put things out there and, you know, tick the boxes. The
Hmm.
moment somebody comes up with a metric
Hmm.
for anything, for any type of productivity,
Hmm.
that metric is very easily abused. It's like GPA, right? So
Hmm.
you're in
Yeah.
school, right? You have a
Yeah.
GPA.
Hmm.
You know how easy it is to abuse the GPA. Like
Yeah.
GPA, if you beg the instructor for higher grade, what gets rewarded? Kind of like slightly unsavory behavior, right?
Right.
A
Right.
person who is honestly taking a course versus a person who is then throwing a temper tantrum in the professor's office.
Hmm.
Chances are the guy throwing temper tantrum will get a higher grade. Not
Hmm.
always, but
Right.
often.
Right.
The same happens with publication metrics. That's why
Hmm.
the impact factor got inflated. That's why the number of publications got inflated.
Right.
I have a lot of thoughts about that too. How that is,
Hmm.
is it good for science? I don't know. We
Right.
haven't come up with anything better,
Yeah.
but it's not ideal.
So basically, there's a quota to reach.
Ah, it's soft, but yeah,
Hmm.
it's good for you to have papers, and it's not good for you not to have papers.
Right.
And
But
so then
yeah.
it creates these papers on the bottom tier of publication quality, where
Hmm.
the second you have something, you just want to put it out there.
Yeah, it's just like kind of rushed with like minimal effort sometimes.
Well, sure,
Yeah.
you could say that.
Hmm. Um, what do you see in like the next 5 or 10 years as the most like beneficial technology in helping like neuroscience or just like molecular biology in general? Like, do you think like something like AI or machine learning will have any impact in this field, or is it
I
too
mean,
primitive right now?
yes, it will have impact.
Hmm.
I'm not sure it will have good impact.
Oh.
Here is my thoughts on it in general, right? Like
Hmm.
there's many kinds of AI. There is AI that works within a set rules, like
Hmm.
AlphaFold.
Hmm.
I don't know if you guys know about AlphaFold, right? I'm sure you do.
Hmm.
AlphaFold is an example of good AI. It has
Hmm.
its limitations, but essentially it allows us to generate hypotheses that we
Hmm.
couldn't previously.
Hmm.
And it's very, very useful. We use it all the time. It's a fascinating tech. If you know its limitations, it's absolutely fascinating and a lot of fun to work with.
Hmm.
AI, the generative AI, the LLMs that create text
Hmm.
are I would put them somewhere in the realm of like TikTok. Is
Hmm.
it good for us to have TikTok? I don't know.
Yeah.
I've sometimes scroll through it, but I don't think it's a good thing,
Hmm.
honestly.
Hmm.
And yes, it feels like people are banking on this AI to solve all problems and to fix
Hmm.
everything.
Hmm.
I am extremely skeptical about it.
Hmm.
And in my usage of AI, the only times we have had some benefit from it has been with coding,
Hmm.
because it used to be that, when you code,
Yeah.
you need to know the syntax. You have to go to bioconductor every time you run into a
Hmm.
problem. You spend like 3 hours in bioconductor, and then you
Hmm.
find the line that works, and then hopefully it works. And
Hmm.
now with like Claude Code or with chat, you can
Hmm.
sort of figure out how it works. The
Hmm.
problem is that first, usually we already know what works means.
Hmm.
So we have controls and we know the results we expect, sort of,
Hmm.
and we can easily test if it's giving you crap or if it is real, right?
Hmm.
So
Hmm.
if suddenly data doesn't make sense, then we can go back and figure out what went wrong.
Hmm.
And in fact, for every single attempt that works, 10 don't. So it makes
Hmm.
shit up all the time.
Right.
As for like generative AI for writing, that is cancer and should not be used, and
Hmm.
it's terrible.
Hmm.
Obviously, generative AI for like image generation and all that stuff, that is insanity
Yeah.
straight up, right? Like,
Yeah.
do ever use it, it's terrible.
Yeah.
So that I am extremely skeptical about AI. I think it's a niche
Hmm.
tool. I think it's useful for niche scenarios.
Hmm.
It might be interesting for, deconvolving neural networks in the brain, perhaps,
Hmm.
but without a way to validate any of this, ah,
Hmm. So in
no.
general, it's just better to use whatever's up here in the brain and don't
I
rely on AI.
uh, don't expect it to be a magic bullet.
Hmm. Yeah.
Like it's a tool. If you know what you're doing, like
Hmm.
every tool, if you know what you're doing, it's great.
Hmm.
If you don't know what you're doing, like you can have a new Steinway piano in your living room, but if you don't know how to play it, it's not going to help you very much, right?
Right.
So same thing.
Right.
Except Steinway piano doesn't screw up the economy and your jobs and the
Right.
environment as much.
Right.
So that's my take on AI.
Hmm.
As far as technology in general, I mean, I think my I think we're getting into like I don't know. I can't predict, right? If I could
Hmm.
predict the
Right.
future, I would. We had this Russian saying, "Знал бы Фрику, жил бы в Сочи." Like if I knew what's in the card deck, I would have a big mansion in the town of Sochi.
Hmm.
I don't know what's in the card deck.
Hmm.
I think transdisciplinary research will continue expanding.
Hmm.
And I think the niche single method stories are going to become more and more niche and less relevant.
Right.
And I think that people who can leverage connections and different ideas
Hmm.
will get ahead.
Hmm.
Um, for example, if you study neuroscience, that's great. But what is classical neuroscience? Well, it's figuring out how this neurons project to this area and
Hmm.
what
Right.
depolarization here causes here, right?
Hmm.
And I'm talking out of my ass here. Like, I don't know. Okay. I'm not a neuroscientist. But how cool would it be to understand how that changes in development?
Hmm.
What happens inside the neuron, for example, what happens with ribosomes of that neuron, what happens with
Hmm.
gene expression of that neuron, how does metabolic change affect that? What
Hmm.
happens when a microglia cell comes in and throws a bunch of cytokines on that neuron?
Hmm.
So connection to, let's say, inflammation, chronic inflammation, cell communication, not necessarily neuron to neuron, maybe astrocyte to neuron, maybe microglia to astrocyte to neuron. And
Hmm.
figuring all that out will require people with different expertise, right? Not
Hmm.
necessarily, hardcore Pavlovian neuroscientist,
Right.
but
Hmm.
maybe somebody who is like an expert in single molecule imaging, maybe somebody
Hmm.
who can do metabolomics, maybe
Right.
some genome assembly person. I don't know.
Hmm. Right.
Like, very different people need to come together and do stuff together.
Right. I really appreciate your time taking to take this interview. I know you're a very busy man, and I
No, no, no,
appreciate
no
you
worries.
for being very frank and down to earth and very honest about a lot
No
of what
worries.
happens in the scientific community. Thank
Yeah,
you for being here
if
today.
sure, if you're interested in any of this, you have my uh, info, and please let me know.
Hmm.
I'm always happy to yak about this stuff.
Hmm.
No problem.
Hmm. Thank you for being here
Cheers.
today.